Hello, everybody. Welcome back. So, in the last talk, we were looking specifically at the areas in which cells are regenerating or cells are dividing actively in your body, and the three main processes where that takes place. So, in this lecture, we're going to start focusing on understanding what is actually happening during cell divisions. What are the processes that are involved? So it's important to know that, we're not just splitting our cells in half. There are very specific processes that go into preparing for the cell to divide, and then the actual process of division itself. And really, we're going to be talking about a series of phases, and that's how biologists tend to classify the different types and processes that are happening in order for a cell to successfully divide. And we call this the cell cycle. And it's essentially just, again, what I said, the sort of ordered sequence of stages in which a lot has to go right in order for a cell to properly divide. And in a broad sense, we sort of group these into two large phases, one of which is the preparatory phase, and the second that is the division phase. And within those, there's lots of different processes that we're going to be discussing. But an easy way to sort of help distinguish everything in your head is like, okay, there's cells that are preparing to divide, and then there are cells that are actively in division. And understanding the steps that occur in both of those broad categories are going to be very important in terms of understanding mitosis. And really, the end goal of all of this is to understand how one cell becomes two identical cells. And that's what's critical here, is really the creation of two identical cells. So let's start thinking about what needs to happen in order for a cell to create two identical copies of itself. The first thing that really needs to happen is we need to copy all the cellular contents. So we need to copy all the organelles that exist. We have to also duplicate our DNA, which we talked about in our last lecture. And you also have to duplicate the amount of cytoplasm that's within a cell. And again, the end goal of this is that each new cell has the same amount as the original cell. So we're really trying to essentially divide and conquer, if you will. We're trying to duplicate all of the organelles that exist. We're trying to duplicate all the genetic material that exists. And we're trying to maintain the structure through the cytoplasm while we do this. And again, we're dividing these into two broad phases, the preparatory phase and that division phase. So your book has this very nice graphic that I thought I would highlight initially to sort of show what we're talking about in terms of these two broad phases. So your book helpfully colors them in two contrasting colors. The first is basically going to be this interphase section, and that's again the preparatory phase, which contains the G1 phase, the S phase, and the G2 phase. And then we actually have the division phase, and that's taking place where mitosis and cytokinesis are actually happening. And importantly, it also shows what's happening from this perspective of a chromosome, right, which we talked about its structure. So this chromosome is all that tightly wound DNA around those protein histones to create these, eventually these sister chromatids, which are bound together by a very important unit called the centromere. And we'll be talking about that more specifically in the next few slides. So let's look at that largest section of that pie. So where a cell is spending most of its life, and that is in interphase. And there are main phases within interphase. The first is what we call the G1 phase. And you've probably gone over a lot of this information in other previous biology courses, so this is really just a refresher And really what we want you to be able to do in this class is to be able to say, okay, why is mitosis occurring? What is important about mitosis? How is then that used in the greater lens of biology? So I'm not here to sort of quiz you about what specifically is happening in each of these phases. That's something you can always go and read in a textbook. But what I want you to sort of think about is what broad processes have to happen in order for a cell to successfully divide, and what happens if those processes don't happen? What could potentially occur? So keep that in mind as we're going through these next few slides. So in the G1 phase, the cell is growing. So it's creating all that extra cytoplasm, because remember, one of the key parts of this preparatory phase is to maintain the structure of the original cell. Once it does that, we're moving into the S phase, and this is where the DNA replication occurs. So all those steps that we went over in our previous lecture of separating the DNA strands and copying them to make sure that we have two precise copies, and we're creating those sister chromatids that are bound together with that centromere. And then lastly, we have the G2 phase. And that is basically where the cell is preparing for division. So the first two phases are creating extra cytoplasm. The S phase, which is most important there, is DNA replication is occurring, and the G2 phase is really the last preparatory step before actual cell division. So let's look back at this image and understand it in terms of, again, of this pie chart. So interphase is where the vast majority of cells are spending their life. We have this G1 phase where the cell is enlarging, we're getting the cytoplasm that it needs, and we're getting and we're starting to begin to produce all of our different organelles. The S phase, and what's most important in the S phase, is your sister chromatids are forming, and that's how I tend to remember S phase, is S for sister chromatids. So we're having our original DNA, we're replicating that DNA so that we get these sister chromatids that are bound together by that centromere. And then lastly is the final preparatory stage, during which the cell is just preparing, getting everything sort of lined up for actual cell division. When we actually move into cell division, we're talking about the process called mitosis. And you've probably had many lectures before that are sort of talking about mitosis. But the thing that's most important to understand, and really the goal of successful mitosis, is that the DNA, these sister chromatids, are separated, and you get basically the chromosome distribution into the daughter cells, where you have the original DNA and the replica DNA for each of the cells. Because again, this is the instruction manual for all other processes that are taking place in the cell. So the last step of the division phase is cytokinesis, and that's really when we're actually creating the daughter cells. So this is where the two cells pinched off divide into what we call daughter cells, which are essentially copies of that original cell distribution. And each one has a original chromosome from the other. So again, cytokinesis, basically this enlarged cell that has double the amount of DNA, splits into two, and then it contains each of the original copy of that DNA. So in our next lecture, we're going to start to really go up close into mitosis, because again, and you've likely had a lot of this material before in other classes, is we're just going to be spending some time looking at what's happened at each step in mitosis, and that's where we're going to pick up next time. So I'll see you soon.